All right, this is week 6 for our um, genetics laboratory lecture. This is the PowerPoint slides. There will probably be other videos this week, but right now, just go over the PowerPoints with you. We're going to review the crosses along with the complementation crosses. I'll introduce those, and that's what this one's about. WT1 should be completed. That's the first, WT1, that was way back. WT2, you should begin and be completed as soon as possible. And if you're not finished with WT1 and WT2, work on them. Let me know if you're not done or whatever. But you should be working on these as we speak. All right, so here we have a wild type control. You should look at this slide, and basically, of course, we're not doing this in the lab, all right, so you don't have to worry about a lot of these details. The main thing is to know what you should be crossing here, all right, and then you'll have to run them and figure out what your results are, all right, so just make for sure you know what you're crossing. All right, so wild type. All right, and here are our other mutants that we're using to figure out where on your chromosome and the nature of your mutant. All right. So like I said, you should be making these crosses now. Don't worry about it as much. We're not really doing double balancers for like we did before we went online, but you do need to understand the material. So you have to kind of understand what's going on theoretically for the test, but it's we're not asking you to do too much. Shall we say there's certainly no there are no tricks here that we normally would have in the class if you were breeding the flies yourself. All right, W8 writing assignment 1. I'm working on. I'll get to them this weekend sometime. Maybe on Friday I'll have some time to start, and they shouldn't take too long. They only take a few minutes to grade. They're not that big a deal. Now realize writing assignment 2 is due in about 11 days. Tomorrow is the 1st, so you'll have about 10 days to do this one. All right, so don't worry too much about this one. Writing assignment 3 will be introduced next week, so just keep in mind what's going on here and the kind of time frame you've got. All right, genetic complementation. All right, just understand that we're two different alleles, usually at different loci. I've kind of explained that in a different video. And they have different functions. Each provides something that the other lacks. If in flies, two strains with different homozygous recessive mutations that produce the same mutant phenotype, well, then we could produce offspring with the wild type. So understand, let's see, different homozygous recessive mutations. All right, so we're talking about homozygous. All right, so recessive. So that's like little a, all right? And then there would be 2, so maybe there's little b as well. But they're both mutations, and they produce the same phenotype. What this means is maybe they're lack of function. So you normally have a pigment in your eyes. There may be more than one way for you to lose that pigment. So you have a mutation, one mutation, in the sequence of the DNA might make the color disappear. You stop making, maybe it's a mutation in which you stop making the pigment. But there might be a different mutation, all right, that makes you make it in such a way that it's translucent or something, so that it appears as if you didn't make it at all. So those are two different types of mutations that might give you the same phenotype, all right? Or so realize that two different homozygous recessive mutations that produce the same mutant phenotype. So we're just saying that there's more than one way for you to get the appearance of your fly that you have. All right, so just keep that in mind, that such a things do exist. All right, so white eye strain 1 and white eye strain 2. Now here what they're doing is they're trying to figure out if with a complementation, if you have a situation like this, so there's big A, and little b, big B, and little a, and do they produce the exact same fly? Well, you're going to have to work it out and figure it out. So here we go. If you look at this cross, you kind of get the idea of what we're talking about. When you look at these two, if you cross these two, all right, you're going to get this, and it's going to look pretty much the same as the parents. But you cross these two, and these two parents look different, and you're going to get this. So keep in mind what we're doing here. And then what happens when you cross these two? What do you get? So just that's how we're kind of approaching this system, so that you can figure out what letters do what does big A do, what does little a do, what does big B, and what does little b, and then how do they work? Are they sex linked, are they not? Now we didn't do sex linked here, but you can look and see and try to figure out what it is that that these mutations do, and they may do something very similar. Both of these, for instance, give you white eyes. But this one gives you maybe something a little different. You get a different combination than you normally would have, and you can cross them to figure out how this might work. So I'll leave this for you to figure out. I think it's pretty straightforward if you understand the crosses and what's going on. So a complementation cross, all right, black body color, you cross them and you get the wild body type. So you mate males displaying similar phenotypes, all right, and then you examine a bunch of flies and you record the selected progeny with images. So the whole idea is you're going to cross some flies, you're going to get some results, and you record those numbers. That's essentially what you're going to have to do. All right, so remember the order. This is testable material. You have to remember that you have to have virgin females. You mate them with males displaying similar. Notice it didn't say identical phenotypes. And then you examine some flies and record the results.